Hi friends, today I will discuss about mucoadhesive drug delivery system.
What is mucoadhesive drug delivery system? Mucoadhesive drug delivery system interact with the mucus layer covering the mucosal epithelial surface, and mucin molecules and increase the residence time of the dosage form at the site of the absorption. Mucoadhesive drug delivery system is a part of controlled delivery system. Bioadhesion is defined as an ability of a material to adhere to a biological tissue for an extended period of time. In the case of polymer attached to the mucin layer of a mucosal tissue, the term mucoadhesion is used. The mucoadhesive drug delivery offers several advantages for controlled drug delivery. The mucosa is well supplied with both vascular and lymphatic drainage. Mucoadhesive drug delivery is a promising area for systemic delivery of orally inefficient drugs as well as an attractive alternative for non-invasive delivery of potent peptide and perhaps protein drug molecules. Introduction. Since the early 1980s, the concept of mucoadhesion has gained considerable interest in pharmaceutical technology. Combine mucoadhesive with enzyme inhibitory and penetration enhancer properties and improve the patient compliance. MDDS have been developed for buccal, nasal, rectal, and vaginal routes for both systemic and local effects. Hydrophilic high molecular weight such as peptides that cannot be administered and poor absorption, then MDDS is best choice. Muco. Inner layers called mucosa. Inner epithelial cell lining. Covered with viscoelastic fluid. Secreted by goblet cells. Composed of water and mucin. Other components include proteins, lipids, and mucopolysaccharides, electrolytes. Main role is protective and lubricates. Adhesive. Tendency substance to remain adhered to surface. If substance adhere to biological mucosal layers is called as mucoadhesion. What is mucus? Mucoadhesive inner layers called mucosa inner epithelial cell lining is covered with viscoelastic fluid. Composed of water and mucin. Thickness varies from 40 μm to 300 μm. General composition of mucus. Water, 95%. Glycoproteins and lipids, 0.5–5%. Mineral salts, 1%. Free proteins, 0.5–1%. The membrane of internal tract example gut, buccal cavity, eye, nose, vagina, rectum are covered with a thick gel-like structure known as mucin. All biological formulations interact with mucin layer during process of attachment. It acts as a link between the adhesive and the membrane. Mucus is a network of mucin glycoprotein that form a continuous layer that intimately cover the internal tract of body. Types of mucosa. Buccal mucosa. Gastric mucosa. Intestinal mucosa. Nasal mucosa. Olfactory mucosa. Oral mucosa. Bronchial mucosa. Uterine mucosa. General structure of mucus layer. Functions of mucus. Protective, particularly from its hydrophobicity. Barrier, in tissue absorption of the drugs and influence the bioavailability. Adhesion, mucus has strong cohesion properties. Lubrication keep mucosal membrane moist. Advantages of MDDS. MDDS offers several advantages over other oral controlled release systems by virtue of prolongation of residence of drug in gut. Targeting and localization of the dosage form at a specific site. High drug flux at the absorbing tissue. MDDS will serve both the purposes of sustained release and presence of dosage form at the site of absorption. Excellent accessibility. Painless administration. Low enzymatic activity and avoid of first-pass metabolism. These dosage forms facilitate intimate contact of the formulation with the underlying absorption surface. This allows modification of tissue permeability for absorption of macromolecules, such as peptides and proteins. Inclusion of penetration enhancers such as sodium glycocholate, sodium taurocholate and L-lysophosphatidylcholine 9-LPC0 and protease inhibitors in the mucoadhesive dosage forms resulted in better absorption of peptides and proteins. Mucoadhesive dosage forms also prolong the residence time of the dosage form at the site of application and absorption to permit once or twice a day dosing. Disadvantages: if MDDS are adhered too tightly because it is undesirable to exert too much force to remove the formulation after use, otherwise the mucosa could be injured. Some patient suffers unpleasant feeling. Unfortunately, the lack of standardized techniques often leads to unclear results. Costly drug delivery system. Medications administered orally do not enter the bloodstream immediately after passage through the buccal mucosa. Oral mucoadhesives patient acceptability in terms of taste, irritancy, etc. The absorption of mucoadhesive drugs is adversely affected by the presence of food. Tetracyclines, in particular, complicates the administration of this class of antibiotics via the oral route. Mucoadhesive drugs cannot bypass liver metabolism. Example: lipids can directly interact with endogenous lipoproteins, thus influencing blood lipid levels. Ulcer-causing drugs may precipitate ulcerogenic effects due to increase in the residence time. Chances of failure of the system due to improper adhesion between polymer and mucosal layer. Mechanisms of mucoadhesion. The mechanism responsible in the formation of mucoadhesive bond. Step 1. Wetting and swelling of the polymer, contact stage. Step 2. Interpenetration between the polymer chains and the mucosal membrane. Step 3. Formation of bonds between the entangled chains, both known as consolidation stage. Step I. Wetting and swelling step occurs when polymer spreads over the surface of mucosal membrane to develop intimate contact. Swelling of polymer occur because the components of polymer have an affinity for water. Step 2. In this step the mucoadhesive polymer chain and the mucosal polymer chains intermingle and entangles to form adhesive bonds. Strength of bonds depends upon the degree of penetration of the two polymer groups. Step 3. This step involves formation of weak chemical bonds between the entangled polymer chains. Bonds includes primary bonds such as covalent bonds and secondary interactions such as van der Waals and hydrogen bonds. Stages of mucoadhesion. Theories: mucoadhesion. 1. Electronic theory. 2. Wetting theory. 3. Adsorption theory. 4. Diffusion theory. 5. Fracture theory. 1. Electronic theory. Attractive electrostatic forces between glycoprotein mucin network and the bioadhesive material. 2. Adsorption theory. Surface forces, covalent bond, ionic bond, hydrogen bond, and van der Waals forces, resulting in chemical bonding. According to the adsorption theory, after an initial contact between two surfaces, the materials adhere because of surface forces acting between the chemical structures at the two surfaces. 3. Wetting theory. It explains adhesion as an embedding process, whereby adhesive agents penetrate into surface irregularities of the substrate and ultimately harden, producing many adhesive anchors. Affinity to surface in order to spread over it. Ability of bioadhesive polymers to spread and develop intimate contact with the mucous membrane. The wetting theory calculates the contact angle and the thermodynamic work of adhesion. 4. Diffusion theory. Diffusion theory describes that polymeric chains from the bioadhesive interpenetrate into glycoprotein mucin chains and reach a sufficient depth within the opposite matrix to allow formation of a semi-permanent bond. Interaction of both polymer and mucin chains to sufficient depth to create a semi-permanent adhesive bond. 5. Fracture theory. This theory describes the force required for the separation of two surfaces after adhesion. Analyzes the maximum tensile stress developed during detachment of the MDDS from mucosal surfaces. Factors affecting mucoadhesion. 1. Polymer-related factors. 2. Environment-related factors. 3. Physiological factors. 1. Polymer-related factors. I. Molecular weight. 2. Concentration of active polymer. 3. Flexibility of polymer chains. 2. Environment-related factors. I. pH of polymer, substrate interface. 2. Applied strength. 3. Initial contact time. 4. Swelling. 3. Physiological factors. I. Mucin turnover. 2. Disease state. Types of drug delivery systems. Depending upon the route of administration of the mucoadhesive drugs, they are different types. They are 1. Buccal delivery system. 2. Sublingual delivery system. 3. Vaginal delivery system. 4. Rectal delivery system. 5. Nasal delivery system. 6. Ocular delivery system. 7. Gastrointestinal delivery system. Different routes of targeting MDDS. Bioadhesive polymers types. Mucoadhesive materials. Bioadhesive polymers are of three types. Type 1, sticky upon hydration and help in bioadhesion. Type 2, forming nonspecific, non-covalent electrostatic bonds. Type 3, capable of binding to specific receptor sites on cell surface. Mucoadhesive polymers. They are water-soluble and water-insoluble polymers which are swellable networks joined by cross-linking agent. Characteristic of ideal polymer. Degradation products should be non-toxic and non-absorbable from GIT. Good spreadability, wetting, swelling and biodegradable properties. Optimum molecular weight. Non-irritant to mucous membrane. Form a strong non-covalent bond with mucin epithelial cell surface. Polymer classification. A. According to their source. Natural and semisynthetic. Agarose. Chitosan. Gelatin. Pectin. CMC. Thiolated CMC. HPMC. Hydroxypropyl cellulose. Synthetic. Carbopol. PVA. PVP. Thiolated polymer. Methacrylic acid. Polycarbophil. B. According to water solubility. Soluble. CMC, sodium CMC, HPMC, MC, PVA, PVP, etc. Insoluble. Carbopol, polyacrylic acid, PEG, etc. C. According to charge. Charged. Aminodextrin, chitosan, carbopol, sodium alginate, pectin, sodium CMC, etc. Uncharged. Starch, PEG, PVA, PVP, etc. Mucoadhesive dosage form. Semi-solid. Gels and ointment. Films. Patches. solid. Tablets. Matrix tablet. Bioadhesive microparticles. Bioadhesive inserts. Liquid. Suspensions. Gel-forming liquids. Mucoadhesive dosage form. A. Matrix tablets. A. Monolithic. B. Two-layered tablets. In monolithic mixture of drug and swelling bioadhesive polymer. Bidirectional release and outer side coated with impermeable hydrophobic substances. In two-layered matrix tablets. Comprises an inner layer based on bioadhesive polymer and an outer non-bioadhesive layer containing the drug for a bidirectional release but only local action. In case of systemic action, outer layer is inert and act as a protective layer. B. Patches. Greater patient compliance compared with tablets owing to their physical flexibility that causes only minor discomfort to the patient. C. Films may be preferred over adhesive tablets in terms of flexibility and comfort. An ideal film should be flexible, elastic, and soft, without breaking due to stress from mouth movements. D. Gels and ointments. Advantages over other dosage form is that they are easily dispersion throughout the mucosa. But accuracy of drug dosing may not be as accurate. Certain polymer are used such as NaCMC, xanthan, carbopol, hyaluronic acid. They change from liquid to semi-solid. HPMC has been used as an adhesive ointment ingredients. Formulation design. In MDDS contain the following functional agents. 1. Mucoadhesive agents. 2. Penetration enhancers. 3. Enzyme inhibitors. In MDDS contain the following functional agents. 1. Bioadhesive agents. 2. Penetration enhancers, 3-lauryl ether, A-protenin, azone, benzalkonium chloride, cetylpyridinium chloride, cetyl-trimethylammonium bromide, cyclodextrin, cod liver oil extract, dextrin sulfate, lauric acid, propylene glycol, sodium EDTA, sodium glycocholate. 3. Enzyme inhibitors, A-protenin, bestadin, purimycin, and some bile salts. 4. Solubility modifiers, cyclodextrin. Mucoadhesive agents. The polymer hydration and consequently the mucus cohesive properties that promote mucoadhesion. Swelling should favor polymer chain flexibility and interpenetration between polymer and mucin chains. Example, polyacrylic acid, PA. Polyvinyl alcohol, PVA. Sodium carboxymethylcellulose, NaCMC. Sodium alginate. HPMC. HAC. HPC. Various copolymer of acrylic acid such as acrylic acid polyethylene glycol monomethyl ether copolymer have also been studied. Penetration enhancer. Substances that facilitate the permeation through mucosa are referred as permeation enhancers. It should be safe and non-toxic, non-irritating and non-allergenic. Pharmacologically and chemically inert. They should have no pharmacological activity within the body. Example, benzalkonium chloride, dextrin sulfate, fatty acid, propylene glycol, sodium EDTA, etc. Recently, chitosan and its derivatives, polymers already known for mucoadhesive properties. Chitosan help transportation of drug through paracellular pathway. List of permeation enhancer. Benzalkonium chloride, dextrin sulfate, fatty acid, propylene glycol, menthol, phosphatidylcholine, polysorbate 80, sodium EDTA. Enzyme inhibitors. Drug and enzyme inhibitors improving the buccal absorption of drugs, particularly peptides. Example: 1 A-protenin, 2 bestadin, 3 purimycin. Bile salts stabilize protein drugs by different mechanism, affecting the activity of the enzymes, altering the conformation of the protein. Chemical modification of chitosan with EDTA produces polymer conjugate chitosan EDTA that is a very potent inhibitor of metallopeptidases, carboxypeptidase. Classification of mucoadhesive polymers. Methods of evaluation. A. In vitro/ex vivo methods. Methods determining tensile strength. Methods determining shear stress. Adhesion weight method. Fluorescent probe method. Flow channel method. Mechanical spectroscopic method. Filling liquid film method. Colloidal gold staining method. Viscometer method. Thumb method. Adhesion number. Thickness. Hardness, kg/cm2. Disintegration test. Average weight content, %. Mucoadhesive strength, gm. Swelling index, %. Stability of buccal tablets. Surface pH study. Electrical conductance. Swelling properties. In vitro drug release studies. Mucoretentionability studies. B. In vivo methods. Use of radioisotopes. Use of gamma scintigraphy. Use of pharmacoscintigraphy. Use of electron paramagnetic resonance, EPR, oximetry. Ex vivo mucoadhesion time. In vivo mucoadhesive performance.